Okay, hi. So let's pick up where we left off. So we're still in supergroup SAR. It's a big supergroup. And so we're going to pick up with the last of the subgroups I want to talk about, which is the subgroup Rhizaria. So if we look at our SAR clade on our phylogeny there, we see again, we have the Straminopoles, the alveolates, and the Rhizarians. So now we're in the subgroup Rhizaria. So everybody in the subgroup Rhizaria is unicellular. Most of them are aquatic. And we're going to talk about three subgroups or clades within the Rhizaria. And that's the radiolarians, the foraminiferans, sometimes just called the forearms, and the cercerozoans, or cercerozoans, however you like to say it. So let's talk about these radiolarians. Okay. So these are actually really beautiful organisms. They have these delicate, symmetrical endoskeletons. That just means an internal skeleton. Like we have an endoskeleton as well, although it's got a very different structure and it's made of different materials. They have what are called pseudopodia that radiate from their central body. So a pseudopodia is like an extension of the body that can sort of bulge out, an extension of the cytoplasm that can kind of bulge out and move the organism around. And we're most familiar with talking about a pseudopodia in the context of what we would think of as the classic amoeba, right? We think of this, amoeba, and it can sort of extend this one of its little lobe-like pseudopodia and move itself around that way, or maybe catch some prey. So amoeba is actually kind of a general term for any organism that can use these pseudopodia, these extensions, this ability to kind of bulge out its cytoplasm. And later when we talk about some groups, we will see what we call the tube-like pseudopodia amoebas. But in this group, the amoebas, and again, amoeba is just a general term for an organism who can use pseudopodia, who has pseudopodia, have what we call thread-like pseudopodia. So these pseudopodia are not sort of lobe-like or tube-like. They are thread-like, very thin. We can see them in image A. And in this organism, the pseudopodia are actually reinforced by microtubules, so they're actually stiff as well. And they're really, really helpful for the organism. It helps it stay afloat in the water really well, helps suspend it, and it also is really great for catching food. So a little organism lands on one of those little thread-like pseudopodia and can actually be engulfed by endocytosis, through that pseudopodia. So it's pretty neat, and they're really obviously very gorgeous organisms. So image A shows us an image of the whole organism, and in image B, we are actually looking at a scanning electron micrograph of just the endoskeleton of the organism, and these can be there's tremendous diversity in what these look like. I've heard it compared to snowflakes before, where each one is a little bit different. So yes, and very, very cool little organism. Okay, so let's go to our next group. And our next group are the foraminiferans. So the foraminiferans have little shells as well, but these shells are, instead of being made of silica like they were with the radiolarians, and maybe I failed to say that the radiolarians, their shell is made of, that endoskeleton type shell is made of silica. So these have shells that are made of calcium carbonate. So they're porous shells, and we call them tests. So a test is a shell. So you might see that term coming up in particular in lab. And basically this test is a single piece of organic material that's been hardened by calcium carbonate. So the organism, obviously builds the organic material, and then calcium carbonate can sort of help harden it, kind of in a similar way that the mineralization of our bones hardens them. So accumulated foraminiferans actually make up much of the world's limestone deposits. These are heterotrophs. But they often have a sort of symbiotic relationship with algae that live within their test. Often. So what that means for the foraminiferan then is that they can also benefit from photosynthesis, right? Because those algae living within their shell can photosynthesize and help provide them with the products of photosynthesis. And in return, presumably the algae are getting a nice, safe place to live, and probably the foraminiferan also provides them maybe with some, other food products that the plant cannot easily get, or nutrients the plant can't as easily obtain. So it's a nice little relationship. One thing about foraminiferans that's kind of interesting is that they make an excellent time gauge for the fossil record for aging rock. Because they fossilize really, really well. They're small, they are marine organisms typically, and they're hard. They're hardened with calcium carbonate, so they fossilize really, really well. And so we'll look at some of these foraminiferans under the microscope in lab today. And they also are, as you can see from these images, quite varied in shape. Maybe not as intricately as the radiolarians, although I guess that is in the eye of the beholder. But yeah, lots of diversity in shape as well in the form of these tests. Okay, let's talk about the cercerozoans. So the cercerozoans are amoeboid and flagellated protists that also have the thread-like pseudopodia. So we have, so let me write that down. So what do I mean by flagellated? I mean they have a flagella. So that's all I mean by flagellated. So they have, again, in this group we find in the ones that are amoeboid, having those pseudopodia, it is a thread-like pseudopodia. So thin, not that kind of big bulgy, loby pseudopodia. Most are heterotrophs, although, as you can see in this image, we actually have, you can see a plastid there with some photosynthetic pigments, so there are some that can do some photosynthesis, but most of them are heterotrophs. That are going to feed by catching prey on their little thread-like pseudopodia. In this group, we have parasites, we have some predators, and some members possess a plastid, and plastid is just kind of a generalized term that would include things like chloroplast or chromoplast. In this case, we're talking about something like a chloroplast. It's containing a photosynthetic pigment. So the neat thing about this is that, so some members, and I apologize that I keep sniffing, I have a little cold. Some members have a plastid that originated from secondary endosymbiosis. And one of the neat things about this, these members of the cerciozoans that have this secondary endosymbiosis is that plastid, called the chromophore, that the arrow is pointing to in the image, retains a little bit of the engulfed algae's nucleus. So remember secondary endosymbiosis. So endosymbiosis, just plain old endosymbiosis, is when a eukaryotic cell engulfs a smaller photosynthetic prokaryotic cell. Secondary endosymbiosis happens when a eukaryotic cell engulfs another cell that is eukaryotic and already has a chloroplast or some other type of, plastid. So in this case, this organism, or at least the lineage leading up to this organism, was eukaryotic and engulfed an algae that already had chloroplasts, and or I call them chloroplasts, just some kind of plastid. And one of the evidences of this is that the plastid found in this organism we're looking at actually retains a little bit of the engulfed algae's nucleus. So that algae that got engulfed had a nucleus, and it hasn't quite disappeared yet over evolutionary time. So that's pretty cool. And if that's not strong evidence for endosymbiosis, I don't know what is. So I think that's a pretty neat thing about this group. All right, everybody. We finally made it to our next supergroup. Yay! Supergroup Excavata. All right, yay! And as you can see, from looking at this phylogeny, this is not going to be quite as large as the SAR. So this supergroup is actually right now kind of weakly supported as a clade. I wouldn't be surprised if in the future things get kind of shuffled around. Again, these phylogenies are hypotheses, and as more evidence comes forward, we shuffle things around. And this is really true for protists. They split from other eukaryotes so early in evolutionary time that their exact relationships is kind of a little bit unclear. One thing we see in this group that helps researchers sort of put them together, many lack mitochondria or have some kind of highly modified mitochondria. So they either don't have a mitochondria, or their mitochondria is not normal. They've got sort of an unusual mitochondrial situation. Ooh, mitochondrial situation would be a good name for a band. I'm not a musician, but I like to think of band names. Okay, so another thing we see in this group that gives the group its name is that some have what we call the excavated feeding groove. So that's what we can see in kind of this picture. See how we've got that kind of in the organism, and I know you probably can't see my cursor if I move it around. We have kind of in this sort of center of that organism, you see this kind of excavated groove. That's part of what gives this supergroup its name. So there are 3 groups within the super group Excavata we're going to talk about. The diplomonads, the parabasalids, so that's 1. The parabasalids, and the euglenozoans. Okay, so starting with this gorgeous creature, let's start with our first group in our Excavata, the diplomonads. So these are mostly anaerobic. Oops, haha, just kidding. So they mostly are organisms that do not require oxygen. They have modified mitochondria. We refer to as mitosomes. They typically have 2 equal-sized nuclei. That's a U. Let me just do the word over. Typically multiple flagella. And many are parasites. Including this gorgeous creature, Giardia intestinalis. So this is what gives you the lovely condition, Giardia, which is common among hikers, campers. If you perhaps drink a water source that's not particularly reliable, you could end up with a fair amount of intestinal distress from this organism. And if you've ever had Giardia, you know what that's all about. If you've never had it, be careful about the water you drink, especially if you're not sure about the source. Bring a bottled water. That's my PSA for today. All right. Yeah, this guy's pretty cool looking. Let's talk about our next group, the parabasalids, not to be confused with salad. Don't eat these, although, you wouldn't know it, they're so little. That's not comforting, is it? Okay. So again, we have mostly anaerobic. I don't know why I can't spell anaerobic today. I'm sorry. Ooh, I keep wanting to switch the A and the E. So these also have modified mitochondria. In this case, they are known as hydrogysomes. And what these mitochondria can do, and the reason they get this name, hydrogysomes, is they generate energy anaerobically and release hydrogen gas as a byproduct. So not needing energy. I'm sorry, not needing oxygen. Okay, so they generate hydrogen gas as a byproduct. So this group also includes many parasites. Trichomonas vaginalis, that you are looking at here, is a great example of this. So if you've ever heard of the STD, sometimes colloquially known as trick, this is what causes it. So yeah, this is a sexually transmitted parasite that travels in the mucosa of the vaginal wall and the urethra, feeding off those tissues. It has flagella, and it has a kind of undulating sort of membrane, and it kind of, just works its way around your body, sort of in an undulating sort of flagella-driven motion. Not something you want. So, yeah. I guess that's my other PSA. Watch out for that one. All right, and finally, and this is one that might look a little familiar because in some of our microscopy labs, we've looked at Euglena as kind of just an example for drawing practice. So the euglenozoans. Okay, so I have to tell you that in the euglenozoans, we're going to have two main groups we're going to talk about. So the euglenozoans as a group are quite diverse. We have predatory heterotrophs. We have some that are photosynthetic autotrophs. And we have some that are parasites. So within this group, we have a lot of different strategy in terms of getting a meal. They have mitochondria that contain kind of these unusual disc-shaped cristae. And I don't have a good picture right here. I'm going to try to remember to have one in the next video. And if I forget, remind me in lab and we'll show you a picture of that. And one of the interesting synapomorphies of this group is that all members have this sort of crystalline, and by crystalline I mean made of crystals, sort of a crystal format, have this crystalline rod in their flagella. So the image, so we have an image of the actual, cartoon drawing of a euglenozoan, and they're showing like a cross-section of their flagella, and we have this kind of crystalline rod here they're showing in cross-section that is part of the structure of their flagella. So all members, okay, let me back this truck up. Okay, all members have spiral. So we have kind of a interesting situation going on with the flagella. So the two main groups are the kinetioplastids and the euglenids. Excuse me, that sneeze snuck up on me. How many of you at home are saying God bless you even though I can't hear you? That's something I would do. Okay. All right. So let's talk about this group for a second. So as usual in this group, we got to have something weird going on with the mitochondria because that's how that's how this supergroup rolls. So we've got a single, unusually large mitochondria. Actually, we should say mitochondrion because that would be the singular. In this group, we have mostly parasites, although some are predators on bacteria. Here you are looking at a very famous kinetoplastid known as Trympanosoma. Trympanosoma is what causes African sleeping sickness in humans. It's transmitted by the tsetse fly. It's got two hosts, humans and the tsetse fly. And one of the things that this organism is able to do that makes it particularly difficult for our immune system to fight it is that it is capable of very quickly evolving a new set of proteins on its cell surface. So once our immune system, that's part of how our immune system works, it learns to recognize the proteins on the cell surface of an organism. Well, very, very rapidly, it's able to, in the next generation, come up with a different set of surface proteins. So it makes it very, very difficult for our immune system to keep up with it. And we'll be looking, let me just back up so I can explain this image, in case you're wondering. So, because we're going to look at this in lab, and this is going to be one of the more difficult images to see because this one's so little. But the red, or the reddish sort of circular donut-shaped things, those are red blood cells. So you're looking at a SEM of a sample of blood, and then these sort of kind of purple-gray, squiggly looking, that is the Trympanosoma. So that is what that's the actual protist, and it's sort of among the red blood cells from a human blood smear. So this is not a particularly friendly group. And then finally, let's talk about the euglenids. Which are a little less scary. So the euglenids, one of the things we find in euglenids is at the end of the organism, at one end anyway, there's a pocket that holds one or two flagella. And one of these flagella is, in this case anyway, and in the organism we'll look at in lab, is pretty long and obvious, and the other is quite short and hard to see. And under the light microscope, you're not going to be able to see the little short flagella. You'll only see the long one. One of the cool things about euglenids are many are what we call mixotrophs. That can either photosynthesize or consume nutrients from the environment if the sun is absent. Can photosynthesize if sun present. Can consume So it is a nice kind of dual strategy situation, depending on what the environment is like. Okay, so that's where I'm going to stop with this video. We're going to pick up talking about the next supergroup, Archaeplastida. So stay tuned. It's on the way.